Topic is maternal recognition of pregnancy. MRP. What's MRP? MRP is actually a physiological process. ये एक physiological process होती है in which fetus जो होता है it signals its mother or dam to maintain the corpus luteum and sustain continuous production so that the pregnancy is maintained. अगर MRP अगर ये signaling mechanism नहीं होगा, if there is no MRP, then the maternal system जो होता है it treats cycle as non-pregnant, and the uterus जो होता है it releases prostaglandin, that's PGF2 alpha, it goes to the ovary, it destroys the corpus luteum, and there is no progesterone, there is no pregnancy, so pregnancy is terminated and new cycle begins. Now discuss introduction. Maternal recognition of pregnancy, it was discovered by R.V. Short, and the aim of the MRP is to maintain corpus luteum and thus pregnancy. It occurs before implantation, and uterine PGF alpha is the main luteolytic agent responsible for the luteolization of corpus luteum and termination of pregnancy, and masking of PGF alpha secretion is important to maintain pregnancy. Okay, जो embryo होता है it secretes protein, steroids to signal its presence to the maternal system. Along with embryo, its fetal parts and secretions that includes protein, steroids to signal protein, steroids ये signal करती हैं maternal system को, so that pregnancy is maintained. Continuous endometrial development and secretory activity, these are essential for CL maintenance. And what's general mechanism? Mechanism of, let's discuss first mechanism of luteolysis. What happens in non-pregnant animal, क्या होता है, जो endometrium होता है it produces PGF2 alpha, and this PGF2 alpha via utero-ovarian counter current exchange it reaches to the, especially in case of ruminants, it reaches the ovary, and it causes apoptosis of luteal cell, and finally regression of corpus luteum, and there is no pregnancy in case of, a new cycle begins. There is in case of non-pregnant animals, non-pregnant cycle. While as there is role of oxytocin receptors, important role of oxytocin receptors in the secretion of PGF alpha. Let us discuss first this corpus luteum, that CL present जो ओवरी पे present होता है, it releases oxytocin. ये oxytocin pituitary से भी आता है, and endometrium जो होता है डैम का it exposes receptors, इनको हम कहते हैं oxytocin receptors, OTRs, oxytocin receptors, and binding of oxytocin जो ओवरी से आया with the oxytocin receptors, it causes secretion of, it causes release of arachidonic acid. Arachidonic acid is important substrate for cyclooxygenase pathway. Cyclooxygenase pathway leads to the formation of PGF2 alpha synthesis. So we can say that binding of oxytocin and oxytocin receptors cause release of PGF2 alpha, and this PGF2 alpha, when it reaches the ovary, it causes more and more release of oxytocin. So there will be more and more release of PGF2 alpha till luteolysis is complete. So here uterine PGF alpha is responsible for CL regression if pregnancy does not occur. And important here is filamentous embryo is lutotrophy, that it elongates to occupy the uterus and prevents PGF2 alpha secretion. Aim here is to prevent PGF2 alpha secretion so that pregnancy is maintained. Mirror में क्या होता है, embryo जो it stays spherical up to day 19, so no elongation होती है तब तक, and protein steroids from the embryo they block PGF2 alpha release, that's anti luteolytic. MRP in various species. MRP, that's maternal recognition of pregnancy, in various species occurs at different time periods, and it occurs before implantation. So in case of cattle, it is 16 to 17. Cow, in case of 16 to 17 days, and in case of sow it's around 11 to 12 days, in ewe it's 12 to 13 days, and in case of mare it's 14 to 16 days, and in case of goat it is 17 days. In canines and felines there is no MRP, we will discuss later. In ovines, interferon tau released by the mononuclear trophoblast cells, also called ovine trophoblastic protein IFT, it's a 172 amino acid. It is produced between days 12 to 13 of the pregnancy. What it did, it reduces PGF2 alpha release and increases PGE2 synthesis. जो PGE2 होता है it's anti luteolytic होता है, antiviral होता है, and immunomodulatory होता है. ये inhibit करता है estrogen and oxytocin receptor transcription. Oxytocin receptor transcription अगर नहीं होगा, there will be no release of PGF2 alpha and there will be no luteolysis. So it is needed for MRP in EU, that's it binds with oxytocin receptors in the endometrium mechanism मैंने आपको already बता दिया है. And it critical threshold जो होता है that's 5 pulses होनी चाहिए per 24 hour PGF alpha in luteolus induce करने के लिए in case of EU. उसके बाद bovine is the responsible factor is for MRP is BTB, that's bovine trophoblastic protein, or BIFT, that's bovine interferon tau produced by trophoblast cells. Mechanism here is it suppresses expression of oxytocin receptors gene in the endometrium, which decreases PGF2 alpha secretion. The main role, as I already said, is to mask the activity of oxytocin receptors which are responsible for PGF2 alpha. Since the same happens here, EPSI is actually endometrial prostaglandin since inhibits it the linoleic acid released by endometrium of cow, pregnant cow especially. It inhibits PGF2 alpha synthesis. And COX2 inhibitors such as meloxicam जब हम देखते हैं फ्लूनिस मेग्लिमाइन होते हैं, ये प्रिवेंट करते हैं PGF2 alpha synthesis को, because COX2 pathway is important for the synthesis of PGF2 alpha. Prostaglandin E2 from endometrium has luteal protective role. It's immune modulatory, it's anti-luteal and helps in maintaining pregnancy. अब बात करेंगे स्वाइन्स की. स्वाइन में क्या जो important factor है, that's estrogen. It's produced by the developing concept and it signals maternal recognition of pregnancy. ये अक्सर पूछा जाता है कि स्वाइन्स में MRP एजेंट कौन है. Estrogen, and it's produced around 11 to 12 days post conception. At least 2 embryos per horn are required to sustain pregnancy and need sufficient uterine coverage. It's important to mention here that 2 embryos per horn are required to sustain pregnancy. And estrogen does not suppress PGF2 alpha secretion. ये suppress नहीं करता PGF2 alpha, but it changes path. That's it causes its sequestration in the uterine lumen. That it changes its secretion from earlier it was endocrine, now it's exocrine, and it decreases its effect. That's there will be no lutealization and there will be maintenance of pregnancy. So estrogen करता क्या है, it causes sequestration of PGF2 alpha secretion into the lumen. Earlier its role was endocrine, now it is exocrine. In case of mere, MRP signal is migration of concepts throughout the uterine horn. Migration of concepts throughout the uterine horn is responsible for MRP in case of mare, and 12 to 14 times per day between the days 12 to 14 of the gestation. And migration जो होता है, it inhibits PGF2 alpha release. Concept is it secrete करते PGE2 and which वो increase करता है uterine motility को, that's possible MRP signal. And transuterine migration also occurs in mare and sow. In canines and felines there is no MRP. That's in dogs and cats MRP नहीं होता, maternal recognition of pregnancy नहीं होती. Reason, what is the reason of this, क्यों नहीं होता MRP in case of dogs and cats? In dogs and cats ओवुलेशन होने के बाद कॉर्पस ल्यूटियम जो होता है, it remains functional for a fixed period of time whether or not animal is pregnant. चाहे pregnant हो चाहे non pregnant हो, there is corpus luteum. This is because your luteal regression is not triggered by uterine PGF2 alpha in the same way as in ruminants, pigs or horses. This is the reason why there is no MRP in case of canines and felines.